Alright, let's continue talking about cells. This section is the cytoplasm and cellular organelles. So we're going to talk about the structure and function of different organelles, including the ones that are part of the endomembrane system. We're also going to talk about some other ones like mitochondria and peroxisomes and their importance in cells. And we're also going to talk a little bit about the cytoskeleton, so the structural components of the cell. Alright, so inside of a cell are three main parts. First is cytosol. This is a kind of jelly-like substance inside of the cell. This is where most biochemical reactions are going to take place, so a lot of different functions of a cell that are happening, they all need somewhere to occur, and they are usually going to be happening in the cytosol. Next is organelles. These are miniature organs, basically, so these are very specialized structures that each have a different job. We'll look at some of these. And then the cytoplasm is the collective name for both the cytosol and the organelles together. So the internal part of a cell is the cytoplasm, which includes both that jelly-like substance and all of these mini organs. So this is a diagram representing the general human cell, and so we've got our plasma membrane out here around the outside, and then inside of it is all cytoplasm. All right, so cytoplasm again is going to include the cytosol, which is this kind of fluid. We don't really have, an obvious representation of that, but all this kind of yellow-like substance back here that is representing the cytosol. And then we've got a lot of organelles and other things going on here. And so we are going to talk about the endomembrane system, which is shown right here in this area of this particular cell. And then we've got some mitochondria we'll talk about in a little bit, lysosomes, peroxisomes, stuff like that. And then we'll talk about these little string-like substances in here a little bit later. Those are some of the structural supports for the cell. Alright, so the endomembrane system, endo means inside, membrane is a membrane. So this is a group of organelles that work together to perform various jobs inside of the cell, specifically producing, packaging, and exporting certain products that are produced by the cell that need to get out to other areas of the cell or just outside of the cell in general. The endomembrane system is composed of three components: the endoplasmic reticulum, this is abbreviated to ER commonly. Then we have the Golgi apparatus, and last but not least are lysosomes. So the endoplasmic reticulum, this is a system of channels, and it is called continuous with the nuclear membrane, and that just means that it is basically an extension of the membrane surrounding the nucleus. We'll talk about the nucleus in the next video because it is a super unique organelle and a very important part of the cell. We have two types of endoplasmic reticulum. We have the rough ER and smooth ER. Rough ER is named rough because it has a bunch of ribosomes stuck all over the surface of the membranes, and its main job is synthesis and modification of proteins. Proteins are really important. They run our body, so we need lots and lots of proteins. And then the smooth ER is called smooth because it does not have those ribosomes embedded in the membrane. Its main job is to synthesize lipids or fats. It's also going to help store and regulate calcium inside of the cell. Calcium is a really important ion for us to have for various reasons. And it's also going to help metabolize some carbohydrates, as well as assisting with detoxification, which is breaking down certain toxins. So we have a lot of smooth ER in liver cells, for example, because our liver is the main site of that detoxification process. So this is kind of what that endoplasmic reticulum looks like. So we have the nucleus up here, and you can see directly connected to that nuclear membrane right here is the endoplasmic reticulum. So it is continuous with that membrane, and you can see all of these little dots are representing ribosomes on the rough ER, and then when we get to the smooth ER, we do not have those ribosomes present anymore. All right, and again, ribosomes are helping with protein synthesis, so creating lots and lots of proteins, and then the smooth ER helps synthesize lipids, as well as helping with that calcium storage and regulation, and metabolizing carbohydrates and toxins. All right, so very, very important jobs happening on these organelles. The next structure of the endomembrane system is the Golgi apparatus. Its job is to sort, modify, and ship off products that come from the rough endoplasmic reticulum, so proteins. There are two sides, the cis face, this is what is receiving products from the rough ER, and then the trans face, which is the release of materials and vesicles. Cis and trans just refer to something, cis is the same side, trans is opposite. Right, so that's why it has those two names. So the same side as the rough ER and the opposite side of the rough ER. So similar picture to what we just saw. We've got nucleus up here, rough endoplasmic reticulum over here with the ribosomes, and then we have our Golgi apparatus out here. So the rough ER is going to send these little packages to the Golgi apparatus, to that cis face of the Golgi apparatus, and they are going to travel through the Golgi apparatus to get packaged up and ready to release off the trans side and moved to either elsewhere in the cell or sent out of the cell as needed, depending on which substance is being produced and where it needs to go. Third and final part of the endomembrane system are lysosomes. So these are vesicles, little packages that have a bunch of enzymes in them that break down unneeded cellular components like a damaged organelle that is no longer functioning and not helping the cell any. So we need to get rid of stuff like that to keep the cell clean. Lysosomes are responsible for two processes, autophagy and autolysis. So autophagy is self-eating, so a cell will basically eat similar cells, or it can kind of eat itself in ways. Autolysis is essentially self-destruction, so the cell will burst itself apart. Subtype of this is apoptosis. The definition of apoptosis is programmed cell death, which means that basically cells in our body recognize when they are getting basically too old and are not functioning as well as they should be any longer. Many cells can also recognize when they have been infected by different pathogens and know that the best thing for them to do is basically to self-destruct to prevent those pathogens from spreading, or self-destruction because they're old and no longer productive to the human body. So best thing to do is break those down and reuse all of the components, making them up. Next, we'll look at the organelles that are for energy production and detoxification. So mitochondria, the powerhouse of the cell. Most of us have learned that at some point in our life. Mitochondria are the site of cellular respiration, which is the creation of energy, so breaking down glucose and transforming it into energy storage systems, and we need energy to run our bodies. So mitochondria are very, very important. I would not call them the powerhouse of the cell technically, or the most important organelle in the cell, because without everything else, then the mitochondria are kind of useless in a lot of ways. Some cells are going to have more mitochondria than others, so muscle cells, for example, they're going to have lots and lots of mitochondria compared to, say, a cell lining the outside of the bladder, because muscles obviously are going to need a lot more energy than the cells on the outside of the bladder are going to need. Mitochondria do have a double lipid bilayer membrane. I've got a picture of that here in just a moment. And then peroxisomes, their main function is to assist with that lipid metabolism, so breaking down lipids and then other chemical detoxification. Basically, this is done by transferring hydrogen atoms to oxygen, creating hydrogen peroxide in this particular case, not water. So moving hydrogen atoms around, we won't get into all the chemistry that this is based on, but moving hydrogen atoms around can help detoxify a lot of substances. So this is what a mitochondria looks like. We have a graphical representation on the left and then an actual image of a real mitochondria from a cell on the left. This is a microscopic image. And so you can see we have this outer membrane space and an inner membrane space. Those two layers of that membrane. So that second layer is what's creating all of these little canals and folds inside of the cell. They're called cristae. And so that is that double membrane system. And in here is where all of that ATP, all of that energy is being produced, is inside of these. And we need a lot of surface area, and that's what that membrane provides. And then peroxisomes, these are other little packages similar to the other types of packages we've talked about. Peroxisomes, again, are going to help break down lipids as well as detoxify different substances. And we do need enzymes to do that to help speed up these processes. That's what enzymes do, is speed up reactions, chemical processes that would otherwise take a very long time. And so peroxisomes also have a lipid bilayer, so that phospholipid bilayer. And then there's a crystalline core inside that is containing all of those enzymes that they will release to help start breaking down lipids and toxins. Then we are going to talk about the cytoskeleton. So this offers structural support just like our bony skeleton does, so we need structural support for our cells too. The cytoskeleton can also help with some cell motility or movement of the cell. They're also essential for reproduction of the cell and then moving substances around in the cell and kind of helping to direct those different packages where they need to go. So cytoskeleton is made up of a variety of fibrous proteins. So we have microfilaments, intermediate filaments, and microtubules. So microfilaments are made out of a protein called tubulin, and these are going to help maintain the cell's shape and structure and resist the cell getting squished. These are also going to help anchor and position organelles within the cell and keep them from moving around too much, because a lot of them we want to keep close by to each other, like that Golgi apparatus. We need it to stay close to the endoplasmic reticulum. Centrioles are a subtype of microfilaments, and these are the origin point for microtubules. Intermediate filaments are made out of keratin. Keratin is the same thing that our nails and hair are made out of. And then microtubules are made out of a protein called actin. We'll talk a lot about actin when we go over muscle contractions. It is really, really essential for muscle contraction, but also in the cell for reproductive purposes. So looking at this diagram again, these little hair-like structures, we have a lot of different types of structures. So intermediate filaments are represented in purple here. Then we've got microtubules here in these kind of thicker tan branches, and then microfilaments are represented here in green. And then we have this centrosome here. The centrosomes are also part of the cytoskeleton, and these are what's going to attach to chromosomes during cellular replication and division, and they're going to pull the two halves of chromosomes to either end of the cell. So we really need those centrosomes so that they can attach to those chromosomes using some of these filaments and microtubules in here. We'll talk more about cell reproduction in another video. So we've got, again, microscopic images of these different structures. So these first two images over here, this is something called fluorescence microscopy. So we add fluorescent dyes into the cells and we can look at them using special microscopes. So we can use different colored fluorescent dyes to highlight different structures. In this case, we have the tubulin protein here represented in this green structure, so you can see how much of that tubulin protein there is present. And if you remember back here, tubulin is what is making those microfilaments, and again, they are really important for maintaining cell shape and structure, as well as helping to get organelles in the correct position within the cell. And so it makes a lot of sense to have a lot of this protein inside of a cell. Next here in red is actin, the actin subunits, so not quite as many of these in this particular type of cell. There would be a lot more actin in a muscle cell, for example, because it's so essential for muscle contractions, but we do still need that actin present. And then last but not least is this keratin protein. This is what keratin looks like under a microscope. So all of these little layers twisted in on top of each other. And that ends our second video for Chapter 2.